In 2013, researchers at Princeton and Columbia estimated that the average US person knows about 600 people. Meaning, all things being equal, if the devastating 1918 flu that blazed through 5% of the world's population were to happen today, the average US person would have known 30 people who passed due to its destructive power. That specter of humanity's past evolved to become one of the most dominant strains of seasonal flus we still have going around today. You know, the one that knocks you off your feet for a week, but is ultimately survivable. And while much of its sheer destructive power has been tempered with time, the seasonal, cutesy flu still kills up to 500,000 people a year, putting it on par with prevalent threats like malaria. As of the time of me writing this, the world scientists have been on edge with regards to one type of flu in particular, the H5N1 bird flu. Historic outbreaks of this flu have had a case fatality rate of around 50%, putting its lethal potential on par with the terrifying Ebola virus. I'm going to give you a brief overview of how influenza viruses work, taking you from the very basic molecular biology of how the flu virus operates all the way to understanding what makes the H5N1 virus so uniquely terrifying. And of course, we're going to dive into what some of the smartest scientists on the planet are doing to prevent this specter of our past from rising again. But before we get into the video, a word of caution. I can tell you how the flu virus works, but none of this is easily translatable to medical advice. Go to your doctor or trusted healthcare communicator for that info. With all that out of the way, let's begin. The problem with studying the flu virus is that the flu isn't just one virus. Influenza is a huge family of related viruses, not of all which have relevance for human health. The flu viruses we care about belong to the type A classification, and even within this nomenclature lies an awful litany of subclassifications. See, part of what makes the flu virus so terrifying is its ability to mutate, something that we'll talk much more about later in the video. Real life isn't Plague Inc. Mutations don't simultaneously update every virus of the same type all at once. Instead, at any given point of time, there are countless kaleidoscopes of local strains of the flu all extant at the exact same time, which is why researchers have adapted this particular nomenclature for each strain of the flu, a categorization that takes into account the locality of each virus. Notice how in this classification scheme that host species is not included. This is because many flu viruses are pretty casual about which host they infect. When the media called the 2009 influenza pandemic the swine flu, it wasn't because that flu only infected pigs, it's because an existing strain of human influenza happened to mix genetic material with the strain of pig influenza, giving the virus a new lease on life and a freshly minted license to kill. So if the influenza virus is in fact kind of casual about what animals it can infect, how come some flus seem to affect some animals worse than others? A major determinant of this virus's host species specificity has to do with this part of the classification system that describes the viral coat proteins. When scientists describe the flu, they usually shorthand this messy classification system with just this part, the H number and N number. These two letters describe the type of protein the flu exhibits on its viral coat. Our seasonal flu, our 1918 pandemic flu, and the 2009 swine flu are all separate substrains of the H1N1 virus, a flu virus making hemagglutinin type 1 and neuraminidase type 1. It follows then that the H5N1 bird flu is making the H protein type 5 and protein type 1. In the diverse family of influenza A viruses, there are at least 18 H proteins and at least 9 N proteins. The viral coat protein hemagglutinin is responsible for the attachment of the influenza virus to whatever cell it's infecting. It binds to sialic acid receptors on the host cell, which can be shaped differently in different host species. H1 through 3 seem to be pretty well adapted for both humans and pigs, whereas domestic birds are infected with a much greater diversity of influenza viruses. While the highly pathogenic avian flu is an H5 virus, making a type of protein that does not bind to human cells very well, successful infection of a host species is a composite metric. It does not rely solely on how tightly that receptor binds, but a combination of everything a particular virus has to offer. But the fact that this virus doesn't seem to be that amazing at binding human cells might explain that, at the time of me writing this script, that cases of the current bird flu happen to be quite mild. And while the thought of the highly pathogenic bird flu H5N1 being like cheap Velcro to our cells might offer a bit of comfort, the reason why scientists continue to be so nervous with regards to this virus is because of a phenomenon known as reassortment. Reassortment is a feature of viruses with segmented genomes, like what the influenza virus has. The influenza virus doesn't just have one piece of genetic material, it has 8 that act kind of like viral chromosomes in a way. If two viruses of a slightly different subtype happen to infect the same cell, along the process of viral assembly, some of those chromosomes can end up mixing. What you have now is a brand new virus with genetic sequences from both original viruses. It is hotly debated as to whether or not the 1918 pandemic flu that took 5% of the world's population exploded onto the scene due to a reassortment event. But what is known is that the 2009 swine flu pandemic was. Several reassortments between human, avian, and swine flus gave rise to a superbug, a fresh face our immune systems haven't seen, and this novelty had devastating consequences for an estimated 280,000 people in the 2009 flu season. The current H5N1 pandemic virus arrived on the scene in 2020 as the result of several reassortments from several different flu strains. This Frankensteining of the flu supercharged its ability to decimate bird populations, especially domestic poultry, a major contributing factor to the current increase in egg prices. While this virus is absolutely ripping through birds, it seems to be pretty mild in humans. But wait, didn't I say that H5N1 had a 50% case fatality rate? It isn't entirely clear why the current strain of H5N1 circulating in North America has a much lower case fatality rate than all the other H5N1 outbreaks. There are some hypotheses that failure of robust testing of mild illnesses in those other cases may have artificially increased the apparent case fatality rate. This is somewhat to be expected. Mild cases of the flu don't exactly warrant the cost of testing unless an outbreak is huge. So historic outbreaks of H5N1 might have been less deadly than data would suggest, which is optimistic. This is why this video ended up in development hell for months. I wanted to give you all an idea as to whether or not the current H5N1 avian flu is as serious of a threat for humanity as what some scientists are claiming. I realized that there is a fundamental uncertainty in influenza studies, because a pandemic strain of H5N1 that is ripping through humans does not currently exist. And so, providing you concrete answers to a future virus is something I'm not comfortable with doing. I'll say this though, reassortment means that some flu viruses are just a hop and a skip away from being the next plague. If this current 2020 H5N1 virus picks up something that makes it easier to infect, replicate, or spread between humans, that could spell catastrophe. Because if the true case fatality rate for the bird flu is even 1%, that puts it on the same level of measles and polio. Only we have, for the most part, vanquished those viruses with the power of vaccines. For reasons we'll cover later in the video, however, this is much harder to do for the flu. Here's a quote by Jacob Lemieux. "I think we are living next to a volcano and it may erupt or it may not, but we need to prepare for the possibility of a pandemic." It's hard to know whether H5N1 poses a threat to humanity because if the flu is anything, it's predictably unpredictable. But that doesn't mean that scientists are powerless to stop it. By learning about how strains of the flu operate at a molecular level, scientists can engineer weapons to fight against one of humanity's most dangerous foes. We'll start with something that all relevant influenza viruses have in common. These viruses are known for their lightning-fast replication cycle, which allows them to blitz down the defenses built up by your immune system, a strategy unlike any other virus I've covered thus far. Let me explain. When I talked about the rabies virus, I imagined a terribly slow infiltration of the body, with the virus tiptoeing around the immune system, taking days, weeks, maybe even months to reach the brain to really start causing havoc. When I talked about Ebola, I imagined something of a spec ops unit, neutralizing key immune cells before an explosive invasion. Influenza hits fast, and it hits hard with a typical incubation period of just 2 days. In fact, speed is such an important part of the flu's game plan that it has been shown that influenza viruses that replicate faster have worse clinical outcomes. At least that's true for H5N1. Us cells of the innate immune system are no joke. As much as my videos make it seem like the immune system is constantly being trashed by pathogens, nothing could be further from the truth. We win battles every day, and yet our efforts go unsung because when we win, you feel no different. See, the innate immune system is like a militia, ready to engage in combat in just minutes to hours, with the higher tech adaptive immune system taking much longer, days to weeks even. But even with our quick response, the speed at which the influenza viruses replicate is just too much to handle. Amongst the first to respond to an influenza attack are the alveolar macrophages. These specialized macrophages are more lovers than fighters, and were evolved primarily to keep the lungs free of debris. kind of like janitors. As a result, these macrophages are quieter, less capable of strong inflammatory signaling, and not suited to fight off tricky pathogens. But when it comes to an influenza infection, these alveolar macrophages unexpectedly lay down their mops in exchange for firearms. In true militia fashion, these alveolar macrophages respond aggressively to invasion, sending out interferon signals that set the antiviral response in motion, calling in immune reinforcements and acting as shields for the vulnerable lung epithelium. The alveolar macrophage is capable of stifling the viral replication cycle in what is known as an abortive infection. This means that alveolar macrophages can act as a kind of sponge for influenza, soaking up virus and shutting down their replication, shielding the delicate tissue of the lung. This valiant defense by the alveolar macrophage enables reinforcements to arrive. Neutrophils arrive to the battle in staggering numbers, helping to keep the spread of virus under control. Not only are neutrophils useful for containing influenza, it has been shown that neutrophils lay a line of signal flare molecules called CXCL12 that help guide cytotoxic CD8 T cells to the field of battle. That is, of course, whenever these lazy bozos get off of their schloungers and actually begin to fight. Yep, any day now. NK natural killer cells also play a major role in influenza infection, but because this video is going to be long enough as is, why not subscribe to this channel and keep an eye out for a live journal club reading where we can explore real cell and molecular biology data together. It's a super fun aspect of my channel, and I love your reactions to the science that I cherish so dearly. But back to the action. After a couple of days of fighting, the immune system is finally ready to deploy the big guns of the adaptive immune system. Cytotoxic T cells follow the trail left behind by neutrophils to the site of infection and begin destroying infected cells, denying the influenza virus a place to replicate. These cells learn what the influenza virus looks like and start pumping out antibodies to trap, contain, and block the virus from getting into more cells. And that's all well and good, except that in the case of the 1918 flu and the highly pathogenic H5N1 avian flu, this response by the adaptive immune system is too little, too late. The preceding battle between macrophages and neutrophils against the influenza virus is not pretty. Alveolar macrophages end up dying en masse, and while neutrophils are spared from influenza infection, persistent inflammatory activation of neutrophils actually worsens disease progression. Not only that, but the neutrophil's best weapon, DNA sticky nets, end up causing increased collateral damage, tearing up the lung and leaving the lung vulnerable to a secondary bacterial infection. The surprising thing about the 1918 flu, the single deadliest virus in human history, is that the virus itself was not the ultimate cause of death for the infected. It was bacterial infections of the lung. Opportunistic pathogens who are generally not a threat to the body now enter a damaged lung environment. With alveolar macrophage guardians dead or dying, these small fry now have free reign over the body in a cruel 1-2 knockout. If we want to answer the question of what makes the highly pathogenic avian flu H5N1 so deadly, we have to dig deeper. We have to go down to the molecular level. Once the virus is internalized, it dumps its viral payload into the cell. Viral RNA heads into the nucleus, where viral polymerases make copies to be sent out to the ribosome for coat production. Only there's a problem for the influenza virus. Its RNA can't actually be copied or be made into protein. See, RNA is an incredibly unstable molecule. Back in my good old days as a progenitor cell, I used to joke that if an undergraduate sneezed in a neighboring room, then all of the RNA you were working on would degrade. And that's only a mild exaggeration. There are a ton of RNA-destroying enzymes called RNases littered within the cell. Influenza viral RNA as is would get chopped up and destroyed instantly if it weren't for the deployment of a devious trick. See, host RNA gets around the dilemma of RNA destruction by attaching a chemical group to the end of the RNA called the 5' cap. This 5' cap prevents RNA destruction and allows it to actually survive the trip to the ribosome, where it can then be read to make protein. The influenza RNA does not come with the 5' cap, but it has the next best thing, a protein called PB1 that literally steals the 5' cap off of host RNAs. This process is commonly known as 5' cap snatching. This strategy has two clever payoffs. Not only does 5' cap snatching prevent viral RNA from being deleted, it has the doubled benefit of hampering host protein production, which some scientists hypothesize is one of the ways that influenza dampens the immune response. Can't fight the infection if you can't make your weapons after all. The most interesting anti-host response from the influenza virus comes in the form of a protein that shouldn't exist at all. Researchers were studying the protein fragments infected cells show to T cells to better understand the battle between influenza and host when they serendipitously came across a protein fragment that did not belong to any known protein, host, or viral. If we look at the genome of the influenza virus, we can find 8 separate RNA fragments. However, upon first glance, this mystery protein's code can't be found in any of these genes. It turns out that the actual sequence for this protein is embedded in another one of its genes, the gene that codes for the 5' cap-snatching PB1. But how could one gene code for 2 proteins? Ribosomes are recruited to begin the conversion of mRNA to proteins starting at a sequence of DNA called the consensus sequence. Part of this consensus sequence is the start codon, a sequence of adenine, uracil, and guanine, the area of mRNA that officially begins the stitching together of protein. However, because biology is just weird jiggling shapes bumping into one another, sometimes the ribosome might bind to an AUG downstream of the canonical start codon. If the ribosome starts translation of mRNA to protein from this AUG instead of that AUG, the reading frame of the mRNA gets shifted, producing an entirely different protein. This represents yet another way that one strand of genetic material can give rise to multiple different proteins, the other way being splicing, something that I explained in my prion video. Since this new protein was found to be embedded in the PB1 gene, this protein was named PB1F2. For PB1 frame 2. This frame shift introduces a stop codon, an RNA sequence that pops the ribosome off of the mRNA much earlier, creating a protein that's just about 90 amino acids long, give or take depending on which strain of the flu we're talking about. To compare, the PB1 protein is 752 amino acids long. Don't underestimate this tiny protein because PB1F2 punches far beyond its weight class, as it is one of the core determinants of pathogenicity in dangerous subtypes of the influenza virus. While there are a ton of different functions that PB1F2 fulfills for the influenza virus, we'll talk about just one. PB1F2 is a protein that straight up kills macrophages, monocytes, and NK cells by triggering apoptosis or programmed cell death. How PB1F2 does this is unlike any other mechanism I've seen. And when I learned about how it worked, I knew I could not do this mechanism justice. So I've asked my very talented fellow molecular biology content creator Clockwork to lend a hand. Clockwork, take it away.
Yo, absolutely. Thanks so much, Phi. Hey folks, I'm Clockwork. I tell stories about biochemistry using molecularly accurate animations. This PB1F2 mechanism is wild. Let me take y'all down to Molecular Town, where we're gonna meet PB1F2. Now, like Phi said, we're gonna watch this squirmy little fella do an end run through the cellular machinery of a handful of your frontline white blood cells, setting off the intrinsic apoptosis pathway, otherwise known as the cellular self-destruct sequence. Now, the nat20 rolled by PB1F2 here is incredible, and it all relies on how this protein interacts with your mitochondria. First up, this is just a simulation of what full-length PB1F2 looks like. In real life, PB1F2 is an intrinsically disordered protein, meaning it doesn't really have a consistent form, at least in like normal cytoplasmic conditions, and therefore it kind of flops around like a wet noodle. Give PB1F2 the right conditions though, and it can take on a stable shape that causes maximum mayhem. The bad news for our infected cells is that PB1F2 wreaks the most havoc inside our mitochondria. And real quick, let's set the scene and see precisely where PB1F2 gets down to business in our mitochondria. Sure, mitochondrions like this one are the powerhouse of the cell, but you're about to see that this organelle does so much more. Zooming in real quick, mitochondria have lots of these giant pores on their outer membrane. This one right here is TOM40. Let's go ahead and rotate our view and cut away half of this membrane so we can better see what's about to happen and you can get a better sense of just how beefy this pore complex is. PB1F2 is going to slip through the TOM40 side of this channel specifically, and once they get inside the mitochondria, they're going to cause some truly incredible mayhem. If we pan down beneath the mitochondrial outer membrane, we're going to see where the actual damage here happens. The chief superpower of your mitochondria is the fact they have a double membrane. The mitochondrial outer membrane is there to help enclose and stabilize this inner membrane, which is all sorts of folded in on itself, making little structures like this called cristae. And the mitochondrial inner membrane, or MIM, is all wrinkly like this to maximize surface area. We want all of this surface area so we can just absolutely stuff this thing full of the protein machinery we need to turn food into cellular energy. And if we zoom into just the tip of one of these folds, we can see just how crowded this mitochondrial inner membrane is. We want all this surface area to maximize all the membrane space we have to stuff the MIM with as many complexes like this as possible. Welcome to your molecular ground floor. At the apex of one of these folds, in one of your mitochondria, we find the totality of your cellular respiration machinery. And to give the shortest possible background here, basically your mitochondria convert energy from your food into something cellularly useful by moving electrons across these complexes, your electron transport chain. And all of that is done to pump protons inside the interior lumen of your inner mitochondrial membrane. Those protons are concentrated inside the lumen and really want to get out, for reasons. But they're only able to escape the inside of your mitochondria by going through the little turbine you see here in ATP synthase. Turning that turbine is going to allow this part of ATP synthase to build new molecules of something called ATP, which is like the energy currency your cell does to do, most of the business of being alive. And that's just wild, right? Turns out even you are powered by a turbine at the end of the day. But anyway, we're not here to learn about respiration, we're here to see how PB1F2 totally blows this whole thing up. When PB1F2 slips through TOM40, it's going to have an easy time embedding itself in the mostly negatively charged inner membrane here, which is going to cause a really bad time as more and more of this viral protein shows up. While the bottom part of PB1F2, the C-terminal domain down here, usually can stabilize in this helical shape, once it is sort of influenced by all the negative charges inside this membrane, this shape is going to change and fold in on itself, creating a structure like this. All these new little twists and terms aren't helices anymore. They're made up of a protein structure called beta sheets. But let's keep spelling this out. Because it's not just one of these PB1F2 proteins getting scrambled like this, a whole bunch of them are going to fold in on themselves, and they're going to undergo a process called oligomerization. And again, what you're watching here is just a simulation of PB1F2 proteins folding in on themselves and then aggregating together into a structure called an amyloid fibril. And if you've watched enough Phi videos in the past, you've got all sorts of alarm bells ringing right now, because these are the same kinds of fibrils we see in cute and fun disorders like Alzheimer's, Parkinson's, and prion disease. Which I mean, I don't gotta tell you, those are not super fun diseases to get. Now, influenza damage is not going to get to the same scale as prions or Parkinson's or Alzheimer's, but amyloid fibrils inside your mitochondria, like your molecular ground floor, folks, that's a really bad time. Now, the exact mechanism is hazy here, but these fibrils are going to clump enough together to essentially skewer this inner mitochondrial membrane. Either the whole thing collapses under all the tension caused by these fibrils, or these fibers get big enough to straight up punch holes out of the mitochondrial inner membrane. Either way, this is not good. Now what I'm showing here is PB1F2 fibrils violently punching a hole through the inner membrane here. But it could be that these fibrils just clump together in a way that creates stable channels instead of this more over-the-top visualization. What matters here is two main things. One, our mitochondrial membrane potential is getting disrupted, which is breaking cellular respiration. And more importantly, two, some internal mitochondrial components are going to leak out into the wider cytoplasm. In particular, this small little connector right here is going to get loose. This is cytochrome C. In normal respiration, cytochrome C is a connector in the electron transport chain. They hold onto this heme group with an iron atom in the center, which helps pass electrons from complex 3 to complex 4, allowing both to pump protons into the mitochondrial matrix. Once PB1F2 disrupts that mitochondrial inner membrane, though, cytochrome C is free to float around the cell and carry out its second, way scarier job. Once free to float in the cytoplasm, cytochrome C will find and bind to this guy. This protein is called APAF1. Now, cytochrome C binding here was going to activate APAF1, basically changing its shape a little so it can assemble into its fully activated form. Once activated, 7 copies of APAF1 are going to assemble into this terrifying-looking complex called the apoptosome. The apoptosome, get this, activates apoptosis, or the chemical cascade that cells use to self-destruct. The absolutely existential part of this mechanism is the fact that all of your cells currently have their self-destruct machines assembled and ready to go at all times. They just require activation. Once activated, the apoptosome kicks off a whole cascade of self-destructive mechanisms that cause our poor cell to effectively eat itself from the inside. In short, by poking enough holes in this mitochondria that cytochrome C can get loose in the cytoplasm, tiny little PB1F2 has hacked into and activated this cell's self-destruct sequence. And yeah, that was a lot, so let's run through the explanation here one more time real fast. PB1F2 is going to get inside our mitochondria, then it'll embed itself into the mitochondrial inner membrane. The membrane environment will allow PB1F2 to bunch up and accumulate into amyloid fibrils, which somehow are going to punch holes into the mitochondrial membrane, allowing cytochrome C to get loose in the wider cytoplasm. Cytochrome C is the activation switch for your cell's apoptosome. Activating the apoptosome, you guessed it, causes this cell to completely and utterly self-destruct, which you know is a bad time for you and the cells infected by the strains of influenza that express full-scale PB1F2. I know this F2 protein does like other stuff, but nuking mitochondria like this does confer positive selective pressure for these influenza viruses. Mitochondria, again, are way more than just the powerhouse of the cell. They're also former prokaryotes and conditioned for communal virus defense. They therefore are a first-line immune defense inside your cells, activating anti-pathogenic pathways if they detect something is off. Any flu virus that ends up manufacturing full-length PB1F2 will end up getting just enough of a head start against internal immunity to better ensure their chances of survival and propagation, which not only allows full-length PB1F2 wielders to deal out even more severe disease damage, but also opens folks up to way more secondary opportunistic infections once the flu virus has been dealt with, since a lot of these cells getting self-destructed by PB1F2 are, critical frontline leukocyte types, leaving you completely exposed post-infection. So yeah, it's super messed up, and it's one of the wildest angles of viral attack I've ever seen. Being sick is one thing, but it is just absolutely wild how diabolical some of these viruses like influenza can be. But, that's enough time down here in Molecular Town. Back to you, Phi. I mean, with enemies like this, how are we not extinct, am I right?
Thanks for the help, Clockwork, and be sure to subscribe to Clockwork's channel if you want to see more molecular biology in action. Highly pathogenic strains of the flu actually have very similar structures for their PB1F2 proteins, implying that this protein is a major driver for how bad things can get. And while the mystery of this protein is still being worked out, there is one more finding that keeps me up at night. Scientists have identified a crucial amino acid, an asparagine at position 66 within the PB1F2 protein, that is uniquely important for influenza's pathogenicity. A rare mutation that flips this asparagine to a serine is associated with a much more aggressive influenza infection. H5N1 strains that contain this rare asparagine 66 to serine are capable of escaping from the mouth's respiratory tract, even allowing it to migrate to the brain. What's more terrifying is that this same rare mutation is found in the 1918 pandemic as well. The mechanism by which this single amino acid change enables the flu to be much more dangerous is still largely unknown. Understanding how genetic variations in the flu virus affects pathogenicity is crucial. This enables scientists to monitor wild isolates of the virus for concerning mutations like asparagine 66 to serine. I could go on and on about the molecular biology of the flu, but we'll end with the protein that starts the cycle of infection all over again. The N protein is the second part of the viral classification system and refers to the neuraminidase protein, a receptor enzyme on the viral coat that allows it to cut itself free from the host cell. The fitness of the influenza virus must be balanced between the N protein and the H protein, one being responsible for receptor binding and the other being responsible for receptor unbinding. Too much activity in either direction, and you either get a virus that can't stick around long enough to cells to infect them, or viruses that stick so hard to cells they can never be released. What's concerning to virologists is that H5N1's N protein has been evolving over time. A short stalk region in the N protein is associated with greater viral fitness when infecting chickens. However, what epidemiologists have noted is that this stalk region has been elongating over time, which actually seems to improve its ability to transmit between mammals. It's possible that an elongating N protein may be partially responsible for why H5N1's been able to make the jump from poultry to cows, foxes, cats, and humans. How an elongating stalk region maps to improved transmission in mammals is not well understood, but it does involve improving the virus's ability to penetrate mucus and spread in respiratory droplets. Maybe one of you out there will be the one to crack that code. So, to sum, two important factors that may make H5N1 scarier than our seasonal flu are its unique PB1F2 protein and its neuraminidase protein. There are others, but this video is long enough as is. Hopefully I've convinced you that the bird flu is certainly capable of being a very scary threat if the conditions are right. But I don't want to end the video dooming about this potential pandemic. Scientists have not just been sitting on their hands. The ability for scientists to fight the flu, however, is somewhat limited by the flu's unique biology. See, coat antigens are really good targets to design vaccines and treatments against. Catch the flu virus circulating in the extracellular space and body block with antibodies, and that's a great treatment. Unfortunately, the flu virus has evolved a unique adaptation to stifle humanity's usual tools. Enter error-prone polymerase. Usually, in eukaryotes, the enzymes that copy your genetic material have a proofreading function. This is really important for you as it means that you don't end up passing on mutations in your genetic code in a way that could lead to unhealthy cells or worse, cancer. The viral polymerase that the flu comes packaged with has no proofreading function, meaning that the flu virus could introduce 10,000 new mutations every time it copies its genome. It's part of what makes the influenza virus so persistent and not so easily eliminated, like smallpox and polio were. In most cases, this is a very bad strategy. Not copying your genome with high fidelity leads to a ton of viruses that are made that are just not functional. However, what this virus lacks in proofreading, it makes up for in sheer persistence. Because flu replicates so aggressively, the high error rate is mitigated by the sheer volume of replication. Enough new flu viruses will form with mutations that are not debilitating for its survival. A lot of variability happens in the hemagglutinin and neuraminidase coat proteins. This was famously observed when the 2009 swine flu virus developed a point mutation in the neuraminidase coat protein that made Tamiflu not as effective. This constant shifting of H and N protein genes is what is known as antigenic drift, as vaccine researchers are forced to hit a moving target every year. The fact that H and N proteins mutate so frequently means that the flu vaccine has to be updated constantly. And if we ever have a pandemic on our hands, our lives depend on the ability for vaccine manufacturers to quickly scale up operations in reaction to that threat, because there is no way we can predict what a virus that doesn't exist looks like. If only there was a way to make a universal flu vaccine, one that doesn't care about the ever-shifting H and N coat proteins. If only. In 2024, researchers at the Oregon Health and Science University dared to go where no other flu vaccine has gone. The main principal issue with the current coat antigen-based flu vaccine is that teaching B cells what the flu virus coat looks like is not the best strategy when those very same coat proteins are capable of changing rapidly over time. To address this, the authors of this paper chose to ignore the fickle influenza coat proteins and instead went after the proteins inside the virus, the cap-snatching PB1 being one of them. Helper T cells that live in the lung are trained on these internal proteins, enabling them to detect when cells are infected. These helper T cells can then trigger a much faster, more robust immune response, reducing the body's reliance on B cells to make antibodies. Influenza's coat proteins appear to be fine after multiple mutations, but mutations in the internal proteins of influenza results in non-functional viruses, meaning that by going after the internal proteins, scientists are able to hit a stationary target, a target that does not vary greatly between flu strains. And while this vaccine is not yet available, it's promising that vaccine researchers are thinking about how to outmaneuver tricky pathogens like the flu. It's part of what brings me hope for the future. Quick deployment of a robust, universal flu vaccine could stop a pandemic before it even starts. To some, the flu virus is, I would consider, one of the final bosses of human health. It is relentless in its ability to overwhelm the immune system, and it mutates at blindingly fast speeds. Constant reassortment of its genome and a high mutation rate makes it incredibly difficult to stay ahead of, making influenza a predictably unpredictable threat to humanity. It is because of these traits that monitoring the current H5N1 bird flu pandemic is crucial. While human cases of the bird flu have been quite mild, proactive vigilance is the only way to ensure that we do not have a repeat of 1918. Investment into flu research is also investment into the health and well-being of agricultural workers, who will come in contact with flocks of domesticated birds in far greater volume than any of us will. Keeping them safe and protected keeps us all safe and protected. What can we do in the meantime? Unfortunately, I couldn't find a lot about how science can implement preventative measures against the H5N1 pandemic to scale. Can't design a flu vaccine against a flu you can't even identify, meaning that if H5N1 were to ever gain pandemic status, biotech manufacturing would be forced to react instead of attack, losing valuable initiative. The faster R&D can be done in response to a pandemic, the more lives will potentially be saved. The development of a universal flu vaccine, however, would mean that governments may be able to stockpile effective vaccines before a pandemic even starts, minimizing potential tragedy. I really hope we see that happen. Thank you for watching, and if you liked this video, make sure to subscribe for more cell and molecular biology content. Hey, what's that on my screen over there? No, it can't be, no. I can't go back to the editing cave! You can't make me! You can't! No! We finally did it, y'all. A video nearly 5 months in the making. Thank you, Clockwork, again for lending your talent, and thank you to all my YouTube members whose names you're seeing fly across the screen. Turns out the flu is like really complicated, but I hope it was well worth the wait. Following video should not take nearly as long, and I will see you all in the next one.